My name is Fred Locke, and I head the cellular immunotherapy program at Moffitt Cancer Center. I'm really excited to tell you about how ALLO-501A, a new allogeneic off-the-shelf CAR T-cell therapy, could benefit patients and how it's different from current CAR T-cell therapies. What we found in our recent report and showed at the ASCO Annual Meeting is that ALLO-501A and ALLO-501 can lead to responses and even durable responses in B-cell lymphoma patients. And I'll briefly summarize some of those findings. ALLO-501A is an off-the-shelf allogeneic CAR T-cell therapy. It's from a donor or multiple donors actually, and it's gene edited so that it removes the opportunity for those cells to be rejected by the recipient, and it prevents the development of graft versus host disease. In this case, ALLO-501 and ALLO-501A have a TALEN-mediated knockout of the TRAC locus, which prevents graft versus host disease and rejection. And also there's a deletion of CD52 gene, which allows the use of a second drug called ALLO-647, which is a monoclonal antibody against CD52 to deplete normal lymphocytes, again, a CD52 antibody, but not the CAR T cells that have been edited to knock down CD52. This CAR T cell therapy is pretty remarkable. There's 2 clinical trials testing it out. The ALPHA study tested out the allogeneic CD19 CAR T cell therapy ALLO-501 in follicular lymphoma and large B-cell lymphoma patients. And response rates are 75% and complete response rates close to 50% in these patients, over 40 patients treated on this trial. And this is really on par with response rates achieved with autologous CD19 CAR T cell therapies. Patients get fludarabine cyclophosphamide lymphodepletion and this ALLO-647, which depletes normal lymphocytes, but not those allogeneic CAR T cells after they're infused. And importantly, on an intention to treat basis, really the response rates are the same or almost exactly the same because this is an off-the-shelf product. So patients are deemed to be eligible for it, and then we just go ahead and give the therapy, which is very different than autologous CAR T cell therapy, where we need to collect the cells through leukapheresis, wait that 4 weeks for those cells to be manufactured, and try to keep the patient stable while we wait. In this case, allogeneic off-the-shelf ALLO-501 and ALLO-501A can be used very rapidly. And so this ALPHA study testing out ALLO-501A really produced deep and durable responses in these non-Hodgkin's lymphoma patients with the longest ongoing remission at a year. And one of the unique things about allogeneic CAR T cell therapy, or the important things, is that we could actually not only give a single dose, we could give consolidated dosing, or we could retreat if a patient progresses. And this can be done much easier than autologous CAR T cell therapy, where you have to collect or recollect the cells or manufacture multiple batches from that patient's T cells. In this case, again, we just reach off the shelf and give it again. And so really, if we think about this consolidative dosing or retreatment dosing, we can visualize this as sort of a time to treatment failure, meaning if we treat a patient with lymphoma with this allogeneic ALLO-501 or ALLO-501A CAR T cell therapy, and we can give them a second dose. We can also wait for them to relapse and give another dose. And both of those are really still the same therapy, right? And so if we think about it as a time to treatment failure, really the results are quite remarkable. Patients who keep getting this therapy or get it at relapse, there's a survival rate of 63% at 6 months, meaning 63%, 2/3 of these patients at 6 months have not failed the therapy. So I think that's really important. And this is a subset analysis of those who hadn't got prior autologous CAR T cell therapy. So I think that's important to note, which is that we can continue giving the treatment at relapse or as a consolidation, and that's very different than autologous CAR T cell therapy. And I should mention, ALLO-501A is slightly different than ALLO-501. ALLO-501A is tested in the ALPHA2 study, and the only difference is in the CAR construct in that ALLO-501 has the ability to get rid of the CAR T cells with the use of rituximab. It has if we call it a kill switch, we can get rid of those donor CAR T cells Turns out we don't really need those. And in fact, lymphoma patients have rituximab, get a lot of it as treatment, even before CAR T cell therapy. And so the ALLO-501A is simply the same therapy that doesn't have that susceptibility for rituximab to clear it. So we can use it easier. And the data from ALLO-501, the trial with ALLO-501A or the ALPHA2 study really mirrors what we're seeing in the ALPHA study for ALLO-501. Again, off-the-shelf allogeneic CAR T cell therapy really offers some unique things that we can't get with autologous CAR T cell therapy. We can get the treatment very rapidly to the patient off the shelf. We can treat the patients we intend to treat. And we also can retreat a patient when they need it rapidly at the time of progression or as a consolidative dosing. And the ongoing trials are doing this. The ALPHA and the ALPHA2 study are giving a first dose and then a second dose of the allogeneic CAR T cell therapy 28 days after the first. And we think that will increase the number of durable responses. And we see about a third of patients with just a single infusion of ALLO-501, a third of patients with follicular lymphoma or large B-cell lymphoma have ongoing remissions at a year. And again, with this consolidative dosing or treatment at relapse, we think we can get that up to 50% potentially at 1 year in ongoing remission in patients with follicular lymphoma or transformed follicular lymphoma or diffuse large B-cell lymphoma. So very excited about these early results from the trial and the possibility that we could advance this therapy to something that can hopefully eventually be given as a standard of care to patients with B-cell lymphomas like follicular lymphoma, transformed follicular lymphoma, and diffuse large B-cell lymphoma. Thank you for your attention.